Okay, so yes, today we are going to start our new portion, which is the molecular basis of inheritance, in which we are going to study about I mean, at the molecular level, genetics. Okay, so as we already know, we all have the cells. Cells have the nucleus. Nucleus have the chromosomes. Chromosomes have the DNA, and DNA has the gene. And gene is the basic unit of the DNA, right? And it is responsible for the genetics, right? So it is
Yeah,
gene is the only one which is control the particular character for a specific character. So
yeah.
let's start with the DNA. Okay, so what is the full form of DNA?
hmm
Yes.
Deoxyribonucleic
Yes,
acid.
deoxyribonucleic acid, and it is a nucleic acid, right? Nucleic acid are basically the two, DNA and the RNA, ribonucleic acid and the deoxyribonucleic acid. And you know it very well, DNA is a polymer of nucleotide. I told you.
Nucleotides.
Yeah.
Nucleotides.
And nucleotides are made up of.
Phosphate group, nitrogenous bases, and sugar.
Yeah, very right. You said very right. Yes. So if I talk about the nucleotide, so we have the sugar, okay, and sugar is deoxyribose. Okay, the
Or
name
ribose. Or
ribose is
ribose.
basically for RNA,
Oh,
and
yeah,
the
yeah, yeah. Okay, okay, okay,
deoxyribose is
okay.
basically for the DNA, and we have
Mhm.
the nitrogenous bases,
Yeah.
and we have
Adenine,
one.
guanine. Okay.
Yeah, very good. Yes,
Everything
PO4
is,
3
yeah.
minus. This is the phosphate group, right? So all these
Yeah, yeah.
together form a one nucleotide. So it's a chain of nucleotides, so many nucleotides, and then it will form the DNA. Am I right? So actually, if I talk about the DNA structure, this is how DNA looks like. So this is completely as of now, right now, spotlight I'm showing you this complete structure, this one. So this is known as the backbone of DNA. Okay, and this backbone is made up of sugar and phosphate. So it means if it is made up of sugar and phosphate, so this black color is what? Can we say? So it obviously should be nitrogenous bases.
Nitrogenous bases, yeah.
Okay, because the red portion is the backbone which is made up of sugar phosphate. So obviously this black is nitrogenous bases. Okay, so these nitrogenous bases are, as I already told you, see, more the nitrogenous bases, more is the more is the length of DNA. Can we say that? If I
Say it again, sorry.
if I say I have a DNA, you have a DNA. Suppose if I say my DNA contained 5,000 base pair, and if I say your DNA contained 10,000 base pair, so which whose DNA is longer?
Mine.
Yeah, because you have more base pairs, right? So it means
Mm-hmm.
more is the base pairs here, more is the length of the DNA. This is very simple. So that's why we have certain organisms in which you need to know about these phases. So if I say that there is a virus, okay, you can say it's a kind of bacteriophage actually. Okay, so it's a 174 phage. Actually,
Oh,
it is a
psi.
bacteriophage. Yes. So
Okay.
it has a 5386 base pair.
For
Five
phase or
three
phage?
phage, not phase.
No,
So it's
phage.
actually bacteriophage name. So it contained the 5386 base pair. If I talk about there is a one more the lambda phage. Lambda
What's a base pair? Base pair?
base pair.
Oh, that. Okay, okay,
This
okay,
more the
okay. I
base
see.
pair, more is the length.
Okay, okay. I see.
Okay. Lambda
Lambda
phase,
is written as like this
yeah,
thing.
48502 base pair.
Bless you.
Okay, thank you. Lambda phase, so it's a 48502 base pair. Okay, so it means which phase has a more number of base pair?
The
Phi or
lambda
lambda?
one.
It means
Lambda.
lambda length is longer, lambda length is
Yes.
higher as compared to the phi 174. I guess you have heard about the bacteria also, Escherichia coli.
No, I have not yet.
Actually, there is a very famous bacteria, very common bacteria, and its name is the Escherichia coli. So, if I
Hmm.
talk about this Escherichia coli, Escherichia coli, actually, it is very most common bacteria.
In Most common bacteria in what?
Escherichia coli, and it contain 4.6 into 10 raised to power 6 base pair. So 4.6 into 10 raised to power 6 base pair. So it means it has a lot of base pairs. So its DNA length will be higher or lesser.
Higher.
Yeah. And if I talk about the human being, so human contain 3.3 into 10 raised to power 9 base pair. This we are diploid actually. We contain two sets. So it is basically for the haploid. So 3 into 3 10 raised to power 9 base pair. Isn't it higher? Like it
Yeah,
is the
with
most
ours is
highest.
higher.
So it
Ours
means
is higher.
human DNA is quite larger,
Mm-hmm.
right? Okay.
Yeah.
So it is about the like the number of base pair. Now we are coming to the main structure of the DNA. So if I talk about the structure of DNA, you know it very well. We have the deoxyribose sugar, and if I talk about the nitrogen base pair, so you know it very well. We have the purines and the pyrimidines, right? So
Hmm.
if I talk about the purines and the pyrimidines, so purines means adenine and the guanine. I told you,
Yeah, adenine, guanine,
yeah. And if I talk about the pyrimidine,
cytosine, and thymine.
yeah, cytosine and the thymine, and here if I talk about the adenine and the guanine, right? But pyrimidine
Yeah.
also contain one uracil, na? Okay, so if I talk about here, so I guess you know it very well. There was scientist Erwin Chargaff. So Erwin Chargaff says that adenine always forms two hydrogen bond with the thymine. So of course, thymine is also forming the hydrogen bond with the adenine, and same
Mm-hmm.
thing, guanine form three hydrogen bond with the
Yep.
cytosine, and so cytosine also forming the guanine, and this
Yeah.
uracil it will be formed in the mRNA. This thing is basically for DNA. Okay, but when I talk about the mRNA, so it means your this T will be replaced by U, because in the mRNA there is no T, but yes there is U. So I will tell
Okay.
you that. Okay, don't worry. So it means how many purines we have? We have the adenine and guanine, and if I talk about the pyrimidine, so it is a cytosine, a thymine, and the uracil, right?
Mhm.
Okay, so you should know one thing. Actually, we have the DNA strand, and it actually run in an anti-fashion like this. So actually, there is a naming, like it is representing the bonding structure. Also, if it is three prime, then you will call it as a five prime. If this is 3 prime, then
What?
you will call it as a 5 prime. Actually,
Why?
it is representing the direction actually. It is representing you the direction. 3 prime to 5 prime, 5 prime to 3 prime. Why?
Why?
Because why 3 only? You're saying
No,
that.
no, no. Like, why? Like, what is this?
What is 3 and what is 5?
Like, yeah, but what is the entire? Is that like the double helix strand?
This is the DNA.
Yeah,
DNA
yeah, yeah,
helical structure.
yeah, yeah. But why is it called 3 and 5 prime?
Okay. If I talk about the structure of DNA, which I am going to show you, okay, so you will see actually these are bonded with the 3 and the 5. How? I am going to show you. See, if I show you the complete structure of DNA, so you will see actually DNA like nucleotide are attaching to the like one side and the other side. So it is actually attaching to the 3 and 4. It means bonding sites are 3 and 4. So for this, I have to show you a structure. Okay, just give me 1 minute. Okay. So if you see the DNA, so Irvin Chargaff came and he says that adenine always connect with the adenine.
What? Adenine is thymine.
Yeah, adenine always connect with the thymine, right?
Yeah.
Okay. So now I am going to show you the nucleotides in the DNA so that it can help you. Nucleotide in DNA. So that you can see this structure very clearly, and I can show you how bondings are usually there. Okay,
Hmm.
so can you see?
See what?
Right
Yes.
at the right side? See,
Hmm.
if you see at the right side, so this is the first chain, This is the other chain which is running in a helical fashion, but right now we are showing it like this, not in the
Yeah.
helical. Okay, so this is the first chain. This is the like, can you just see, it is a backbone, sugar and phosphate, I told you. And
Mm-hmm.
inside that there is a nitrogenous bases, black.
ATGC.
Yeah, so it is first strand, it is another strand. Okay, so now yes, you asked me ki sir why it has a 5 and 3. So just tell me one thing. See, actually, if I am going to start the numbering, if I do the numbering, so can you just see, this is the sugar. So 1, 2 and 3, can you just see? So at the carbon number 3 there is a bonding downward, like this is the one nucleotide. So one nucleotide is attaching to the other nucleotide at the carbon number 3 of first.
Mm-hmm.
Yes or no? So this
Yeah.
is the carbon number 3 of first, but this is the carbon number 5 which is going to attach here. uh
Okay.
oh, so it is actually it is representing you the like I'm going to show you like this. Yes, can you just see here?
Mm-hmm.
So can you just see, this is the actually carbon number 5 which is representing. Okay, and
That's the
here,
carbon.
yeah, and this is showing,
C6H12O6. Oh, sugar.
yeah.
Yeah. Okay.
See, this is the carbon number 3 where the bonding is going to take place at the downward side. See,
Mm.
this is one nucleotide. This is
Mm-hmm.
another nucleotide. So one nucleotide is attached to the another nucleotide at the carbon number 3 na.
Mm-hmm.
So it means at the downward side can we say it is 3,
Mm-hmm.
and at the above side we can say it is 5. Same,
Mm-hmm.
this thing will become opposite here.
Yeah.
Opposite. At the carbon number 3 it is, and at the carbon number 5 it is. So actually,
Oh,
it is representing you the like direction. It is
at
representing,
carbon number 5, there's a phosphate present there.
yeah, yeah, actually phosphate there. Yes, exactly. This carbon number 5 there is a phosphate group. Okay, yes. So we have to represent the direction actually. So here you can see something like this is structure, something like this. Okay, so this is
Mm.
completely a nucleotide and they are forming the hydrogen bond. Can you just see, hydrogen bomb are forming the two hydrogen bond with the thymine. Guanine always form the three hydrogen bond with the cytosine, I showed you.
Mm-hmm.
Okay, so adenine, thymine, there is a two hydrogen bond. Guanine, cytosine, there are three hydrogen bond. So you have to remember that. So it is completely a nucleotide, and nucleotide. They are going to attach together. Okay, and they are forming the, yes, they are forming the polymer of nucleotide, and they are forming
Yes.
the DNA. Okay, very simple it is. So actually you should know, if I talk about the purine. So purine are the double ring structure. Can you just see guanine and the adenine? So these are the double ring structure. Can you just see here? Where is? Can you just zoom it?
Yes.
Yes.
Yeah.
So
I
these
see.
are the double ring structure of adenine and guanine. But if I talk about the pyrimidine, these are the single ring structure. And
Primitive.
these structures are very important. You have to make it in your copy. These structures you can't compromise, you have to draw it in your copy. Okay, so before I proceed further, I want to show you the complete structure of nucleotide. Nitrogenous base can be of any type, purine and pyrimidine. So it is a adenine. Nitrogenous base you can make here guanine, cytosine, anything you want. This is the sugar, it remain the fixed, it is the deoxyribonuclease, deoxyribose actually. This is, can you just see
Yeah. Yeah.
here,
Yeah.
at the carbon number 5 there is a phosphate group, I showed you. You were
Mm-hmm.
saying, so 1, 2, 3, 4, 5. So at the carbon number 5, there is a phosphate group. So that's why it was 5 prime and it was the 3 prime.
Mm-hmm.
Got it? So at the carbon, you are getting or not?
Yeah. Yeah. Yeah.
So why it has the 5 prime? Because it is the carbon number 5. And why
Yes.
3? Because at the 3 there is a bonding to the downward side. Another nucleotide.
I
one
understand
one
this.
nucleotide can be attached to the another nucleotide by the carbon number 3, which is the, like here carbon number 3 should be there. Okay, so any doubt in this? These
Nope.
structures are very very important actually, so just make it in your copy.
Uh,
So
C
I
double bond O. C double
yeah,
bond O
C
is what
double bond O is ketone and H2 is a
ketone.
amine. So if you want to see the adenine structure, this is how something look like. And if I talk about guanine something look like this. If I talk about pyrimidine, so pyrimidine something look like this. Okay, CH3, NH2, and C double bond O, C double bond O. So these are the thymine, cytosine, and the uracil. Okay, so purine
Yes.
and the pyrimidines are there. So can we say purine always connect with the pyrimidine, like adenine always connect with thymine, and thymine with
Yeah.
adenine, and uracil we will take it in mRNA place. Okay. So actually, what happens here? There was this scientist whose name was the Watson and Crick. Like two scientists are there. Okay. So these two scientists,
Francis Crick and James Watson.
yeah, Watson and Crick. Okay. So
Francis Crick and James Watson.
yeah, exactly. Francis Crick. Okay. So if I
They
talk about,
discovered the double helix
yes,
thing.
so they perform experiment on the like it was a X-ray experiment, right? So if I talk like they actually proposed a double helix model of DNA, okay, which is
Yes.
based on the X-ray diffraction. X-ray diffraction, okay. So what is the meaning of this X-ray diffraction?
Bless you, bro.
Actually, actually there is a experiment on X-ray basis, and this is how the diagram was looking like. I don't know what is happening today. Okay, okay. So this is how it look like this. So in the X-ray, they showed the like they um in very much detail. Of course, they have the microscope. So X-ray diffraction something look like this. So it is representing your DNA is a single helix or the double helix.
Double.
Of course. So by looking at this is how DNA look like. So because of the Watson and Francis Crick, so they performed their experiment, which is the X-ray experiment, and finally they said that DNA is a double helical structure or a single helical.
Double.
Yes. So DNA is a double helical structure, so that's why we usually make a structure like this. Okay, this is very bad, but yes, this is how it is something look like. And the pairing inside the pairing was given by
What? Crick? I don't
pairing
know.
of nitrogenous bases. It was given by
Double helix?
no pairing of nitrogenous bases.
Mm,
Pairing of nitrogenous bases is basically explained by the Irwin Chargaff. I told you Irwin Chargaff, and he says that like adenine, if this side is adenine, thymine, so this side should be adenine, and if this is adenine it should be thymine. If this is G, so it should be
Erwin
C. If this is
Char
C, if it should be
Guff
G. So
said
Irvin Chargaff says that adenine always form two hydrogen bond with the thymine, and guanine always form three hydrogen bond. These are with the cytosine. Okay.
hmm
So Irvin Chargaff, you had to. That's why Irvin Chargaff also says that adenine and thymine they are always equal. So it should be one. Guanine and cytosine are always equal and should be one because they always connect with each other. Say it.
yes.
So sometimes question is asked there like in the complete DNA, adenine is 20%. So can you find out
Uh
what is the percentage of guanine? Yes, think. If adenine percentage is
80%?
80%,
Oh wait, hold on. 30.
how did you calculate that?
Is that wrong?
No, you are right. But how?
Yeah, it's just isn't it math?
Okay. Adenine and thymine are equal, right? So
Okay, okay,
it will
okay, okay, okay.
become.
I got it. Okay.
Oh, it was a
So
trick.
what?
It was trick. You just guessed or calculate
No, no,
that?
no. Okay. I can tell you how I got it.
Yeah, yeah,
So
yeah.
so obviously if A is 20, then T has to be 22, right? So and then 100 minus 40 is 60. 60 divided by 2 is 30.
Yeah, very good. Completely right. Okay. So 40, and now so everything is left. It is GNC only. So you have to
Yeah,
divide
yeah.
by like 60 to 30, 30 only. Got it now? Okay.
Yes, sir.
If I say that if adenine is 30%, then what should be the thymine?
30% as well?
Not 20.
No, 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 no. No, no,
Because
no. They have to be the same, right?
yeah, exactly. Very good. Okay. So remember, if I ask you DNA is made up of nucleotides. So am I right or wrong?
Polymers of nucleotides.
Yeah, can we say DNA are the two polynucleotide chain? Two
Mhm.
polynucleotide chain, okay? Okay, if I ask you the backbone of DNA is made up of backbone of DNA,
Sugar and nitrogenous base and phosphate. Or sugar and
what?
phosphate. Sugar and phosphate.
Yeah, yeah,
Sorry, but
okay.
yeah, I meant
And
sugar
and,
and
and
phosphate.
inside the DNA there are linings which are joined together. It is made up of
Mhm.
like this.
Nitrogenous bases.
Okay, 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 fine. So these two, like nitrogenous bases, are actually going to attach together. So they are going to attach together by hydrogen bond or something else.
Hydrogen bond.
Yes, because one adenine like attach with the thymine with the hydrogen bond, right? Okay.
Hmm.
So these DNA run with the polarity or not, like anti-parallel polarity. If it is 3, so it should be 5. If it should be 5, it should be 3. If it is
Oh
3, it
wait,
is
yeah.
5. So it has
Yeah,
a
yeah.
so it is showing you the direction na. So this is
Yeah.
known as the like these chains are anti-parallel. Remember. So it is showing you these days like they have anti-parallel polarity. Okay? And these are representing the bond na 3 prime 5 prime. Okay? Got it?
Yeah. Yeah.
Okay, you should know one more thing. This DNA
Yes.
from here to here, complete 1 ton. It is starting
Mm.
from here and from here. This is known as a complete 1 ton. So
It's an
it is
S.
known as the one pitch. One
S.
pitch. Yes. You can say like it is starting from here and you have to terminate here. Okay. So this is
Mm.
known as a one pitch. One
I
pitch.
understand. I understand this. I understand.
And in the one pitch, you have to remember there are total 10 base pairs. 10 base
Mm.
pairs. Like one, 2, 3, 4, 5, 6, 7, 8. So it is going to 10 base pairs, right? So in a one pitch, how many base pairs are there? Total
10.
10 base pairs are there. Okay. So if I talk about like there is a distance, like if I say the complete one pitch distance, it is actually equal to 3.4 nanometer.
Huh?
3.4 nanometer, a complete
But
one pitch distance. One pitch has a distance of 3.4 nanometer. So can you tell me the distance between the two base pairs? If one pitch has a total distance of 3.4 nanometer,
6.8 nanometers.
no.
What?
If one pitch has a total distance of 3.4, so this is one pitch,
Okay.
right?
Yeah.
And it total are 10 base pairs.
Mm-hmm.
This total distance is 3.4 nanometer. So if I am asking you only this distance from here to here,
Would you like a proportion or something?
don't you have to divide by 10? Because if it is
Oh shit, yeah.
total 10, it means it is one
0.34
page.
nanometers.
Yeah, so you have to divide by 10. That's why you are going to get the one part, na? 1/10 part. Okay. So it
Uh,
means can we say one pitch? One pitch
so it's
means
0.34
one
nanometers.
pitch means
Okay.
can we say 10 base pair?
Oh
One
yeah,
pitch
yeah,
content.
yeah, yeah.
So it is
Yeah.
3.4 nanometer. So if I want to find out distance into one base pair is covering how much distance? So of course you are
Hmm.
going to divide it by the 10 unitary
Yeah.
method. So you are going to get
The what?
0.34 nano. Unitary
Oh yeah,
method
yeah,
means
yeah.
if one is of 5 rupees, so of course 5 would be 25 rupees.
Hmm.
Okay. Okay. So it is a 0.34 nanometer. Any doubt here? This is the one base pair. Okay.
Wait,
If, if
oh wait, one base pair. Okay.
if you know, if you know the total base pair of the human, because one base pair covering that distance.
Mm-hmm.
So if you know the total base pair, oh sorry, I
You
just
can
wrote.
calculate the distance of the DNA,
Yeah,
which
very
is so
good.
long, which is
Yeah.
so long.
So if I talk about the E. coli, I told you at the first page. Okay,
Uh,
these are the base pair
uh,
number. Why are you
E.
making
coli.
such?
That's a food disease.
Okay, yeah. So Escherichia coli, like this number of base pair and human have a such base pair number, right? 3.3 into 3 10 given by 9. So we can
Yeah,
multiply it by the distance. So when we multiply
yeah.
by the distance, so E. coli distance we got it is 1.36 millimeter. And if I talk about human, so it is 2.2 meter. 2.2 meter length of DNA. Our our s-
Yeah.
no, you
Mhm.
may sleep. You may sleep.
No, okay.
Okay.
I'm fine. I'm fine.
Okay.
Sorry.
So it's completely fine. Yes, our cell size is very, very small. Okay.
Yeah,
But
but relatively,
our
all of them
DNA is
together collaboratively is large.
this is our DNA. Now tell me, this is our DNA which is 2.2 meter and our cell is
Hmm.
very, very small. So how this DNA can be accumulated in that? How?
Wow.
We multiplied by the that distance and distance is coming to the 2.2 meter DNA distance. So how it can come into a very small size cell, very, 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 very small size cell?
Because there's billions and trillions of them.
No, no. One cell contain that length, not the so many cell.
Wait, what was your question?
Yeah, DNA length. What is the DNA length? 2.2 meter.
2.2 meters. Yeah,
By
yeah.
the calculation, we calculate that.
Yeah, 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 yeah.
But our cell is very small and DNA length is like this.
Oh,
So
yeah,
how
yeah.
this long DNA can accumulate inside the cell,
The
even
double
inside the nucleus?
helix. The double helix.
Still, still, I'm talking about the double helix only. It is the length
Yeah.
of double helix. So how this DNA can come inside this very small cell? Okay, I'm going to tell you.
The
I'm going.
chromatin octamer fibers, it condenses all of them, right?
Yeah, yes, yes.
Yeah, I got it.
Because DNA length is higher, but its thickness is very negligible.
Yeah.
Even if I draw so many thread, you won't be able to see that. You have to see
Hmm.
with the microscope, so its thickness is very negligible. So you can fold it, fold it, fold it,
Yeah.
fold it, fold it, fold it. It will become very, very small, even very, very.
Histone octamer.
Yeah.
Sorry, the
So
name.
all those structures, yes. So can we say actually it is packaging of DNA?
Yeah, yeah,
Packaging of DNA means like there is a condensation.
yeah.
Okay. I told you in the cell chapter this thing.
I remember. I remember.
Okay, so you remember I told you one thing, like the in the prokaryotic cell, like there is no chromosome, okay, no specific chromosome, they contain only DNA because they are primitive, they are not
Hmm.
advanced now. So if they are primitive, so their nucleus is known as a nucleoid.
Nucleoid. Yeah.
Nucleoid means like because they are prokaryotic and they don't have so many chromosomes, so they are primitive one. So in the prokaryotic, nucleoid is just like a thread of the DNA inside the cell. Okay, thread of DNA. But if I talk about the eukaryotic, so in the eukaryotic, if we have a cell, so inside the nucleus, we have a pair of chromosomes. Okay, we have a pair of chromosomes. So how many chromosomes we have? 23 pair.
Yes.
And of course they contain the DNA. So I told you DNA is actually condenses and DNA is wrapped by a histone octamer nucleosome,
Hmm. Yeah.
and these nucleosome are further condensed by the,
Chromatin.
yeah, they form the chromatin, but it is folded by the non-histone protein. Octamer is a
Mm.
histone protein, and when they form a structure then become a non-histone protein which forms the
Hmm.
chromatin. So firstly it is DNA is wrapped around a histone octamer. So this is the histone protein. Okay, Then there will be a further condensation. Then we need a non-histone protein.
Mm-hmm.
And when we need non-histone protein, NHC protein, then it forms the chromatin fiber, which is a
Mm-hmm.
very condensed, and finally it forms chromosome. Okay.
Mm-hmm. Mm-hmm.
So you have to tell me one thing: a complete one nucleosome contain the 200 base pair of DNA. DNA
Mm-hmm.
is wrapping around that, na? So it contains the 200 base pair, and inside the nucleosome there are 8 protein actually. Octane histomer, you remember?
Mm-hmm.
One nucleosome is made up of histone octamer, 8 histone protein. So if total nucleosome contain 200 base pair, so can you tell me one histone octamer contain how many base pair approximate?
Wait, say it again. Your voice cut out. Sorry.
Okay, this is one nucleosome.
Mm-hmm.
Okay, and in the nucleosome there are 8 histone protein. 8.
Yes.
That's why its name is histone octamer.
Mhm. Eight. Octamer.
Yeah. So and there is a DNA is wrap around that DNA.
Yes. 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 Yes.
So from here to here, from here's the beginning and from ending, there are 200 base pair are covered.
Hold
200 base
on.
pair
Give.
of 200
I
base,
can. I can do it. Please give me a second.
yeah. So 200 base pair are covered by a single nucleosome. So one histone protein contain how much?
68. 68 nanometers. Yeah. Right.
One second, let me. Okay. Are you sure?
Yeah.
No, you are wrong. Do it, calculate.
68
68.
nanometers.
What are you doing?
Yeah.
I am saying total 200 base pair are contained by the nucleosome. So how many
Mm-hmm.
base pair will be contained by the single histone octamer? How many base pair?
Oh.
Not the
Oh
length.
shit. 20. Right. Oh, no. 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 Not 20. I can't math. 200 divided by 8. Right. It's 25.
Yes,
Oh.
25. Okay.
I understood the question.
So
Sorry. Sorry.
so it is total
Sorry, mom.
a, it is a histone octamer. This is completely nucleosome. So one histone octamer contain the 25 base pair. Okay. Approximate. So 25, 25, 25, 25, 25, 25 become the 200. Okay.
Mm-hmm.
Getting or not?
Yeah.
Finally, you just got the chromatin fiber here. Chromatin
Yeah.
fiber. So if you're getting the chromatin fiber, so chromatin fiber further condenses to form the chromosome.
Chromatin.
uh,
Yeah.
Chromosome, it is itself a
Chromosome.
chromatin.
Chromosome.
It is itself
Chromosome.
a chromatin. Okay, so firstly histone protein come or non-histone protein come for condensation of DNA? Firstly
Histone.
histone, and they form nucleosome. Then
Yeah.
nucleosome further come, then it form it need.
Non-histone.
Okay, actually you need to remember, do you know motiyo ki mala? Motiyo ki mala means like it's a like women usually wear na, like pearl
Necklace.
neck,
Pearl
not
necklace. Yeah.
necklace, but like
Pearls.
yeah, pearls like this,
Yeah. Yeah. Yeah.
garland you can say.
Okay. Sure.
So nucleosome structure is something like a beads on this string, beads on
Mm.
this string.
Oh
in
wait, is that
in
like for marriage?
yeah, you can say that. Okay, so it's
Wait,
something like
no.
usually women wear it na, like
When they
very
get married.
meditative type, not marriage. In marriage people usually wear a necklace in India. I don't know about much about the USA, but yes, in India they wear the necklace. Okay, this is simple based on this thing. My mom also wear that. Okay, based on this thing, motiyo ki mala you usually say in Hindi, motiyo ki
Yeah.
mala. Okay.
Are they like red or black?
What?
What color are they? The beads?
White, 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 white.
Okay. Interesting. Okay. I yeah.
Should I show you?
Sure.
Okay,
Yeah.
okay.
I think I know what you're talking about, but I don't know exactly.
You have seen that, I know that. Yes, can you just see? I searched in Hindi. Okay. Yes, you will
I can't.
see.
I can't see
Can you
it.
see?
Okay.
Can you see? This
Oh
one.
yeah, 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 yeah. I see
This
this.
is how this is known as the beads on the string structure. Beads means like that white circular.
Yeah.
So this is how nucleosome structure look like.
Oh,
Nucleosome
that's
structure.
what it means. Okay.
Yeah. So sometime in the examination, in the NEET examination, you will see beads on the string structure. So beads on this, you have to remember it is a
Hmm.
nucleosome structure. Okay. And finally, they will help in the formation of chromatin. Got it now?
Hmm.
Okay. So now you have to remember one thing. So sometimes DNA is very condensely packed, and sometimes DNA is like this.
Hmm.
So this when it is condensely packed, I mean when there is a lots of condensation, so this type of chromatin, you will call it as a heterochromatin. Heterochromatin. But such type of chromatin, you will call it as a euchromatin. So just tell me which chromatin is very loosely packed, hetero or
Euchromat.
euchro?
Euchromatin.
What? Can you hear?
Yeah, sorry.
Yeah. Euchromatin is loosely packed, am I right? Loosely. Can you just see it is loosely, very quite open up,
Yeah, yeah.
and it is tightly packed. Now just tell me if
Hmm.
if there is a catalyst, and if
An
catalyst
enzyme.
from the DNA, it want to make mRNA, because from the DNA mRNA usually come. So which one can help in formation of mRNA very easily? Loosely one or tightly one?
Loosely one.
Yes, because tightly is very condensed, very dark, very condensed it is. So which is transcriptionally active? Which one is very active for the?
Euchromatin.
Yes, and it is not so active. It is transcriptionally inactive. Transcription means from the DNA to mRNA. Okay, and it stains dark. You have to remember a line. It stains dark, but it is stains light. You have to remember. So which one is very, like both are chromatin, but which chromatin is very important? Euchromatin or heter?
Euchromatin.
Yes, because it
Euchromatin.
is loosely packed. Okay, so the other regions are tightly packed, very dark. So you will call it as heterochromatin, and those regions which are very transcriptionally more active, so it is euchromatin because it is loosely packed. Okay,
Yeah.
and you usually talk in your native language, Andhra
Hmm?
Pradesh.
Oh yeah, yeah. I can speak that. Yeah.
Oh, cool. Okay,
I speak
okay.
3 languages.
Oh, may I know the name?
Oh yeah, yeah. Telugu, English, and French too.
Telugu, English, and French.
Yeah.
Okay, cool. Don't you get confused in all these three? Like sometimes you are speaking like all the combined, all combined languages.
Um,
No,
not that many
I was
times,
just
but
I was just joking. Okay,
yeah, yeah.
fine.
I see.
Okay,
Sometimes, yeah, sometimes.
okay. So we have the Griffith. Now, actually, we want to know which one is genetic material, which thing is genetic material. So there was the scientist name was the Frederick Griffith. Now see what is happening. He took he took rat. I know I'm drawing a rat diagram very bad. This is a rat, suppose. Okay. And suppose this is another rat. All rat are same, okay? Don't laugh.
Yeah.
So how many rat we have now?
4.
Yes. So actually Griffith was working on the Streptococcus pneumoniae bacteria. Streptococcus
Mm-hmm.
pneumoniae. And this Griffith has prepared the strain. Do you know corona strain? New strain?
Yes.
So same thing.
Yeah.
He prepared the two strain of solution. Two strain he prepared from the Streptococcus. One strain was very smooth colonies of bacteria, smooth colonies, and other strain
Mm-hmm.
is rough colonies, like rough colonies of bacteria. Okay, so he was working on the Streptococcus pneumoniae, na, which caused the pneumonia. So he was working on the bacteria. Its name was Streptococcus pneumoniae. And when he was working on Streptococcus pneumoniae, so he prepared the two solution. First is S type. S type means smooth type, and other is R type. R type is rough colonies of bacteria, rough colonies. So S type is very harmful. S type S type causes pneumonia actually. It is very virulent, very very harmful. But R type is not that harmful, so it is a non-virulent. Non-virulent means which is not so harmful. Okay. So Griffith was working on this Streptococcus pneumoniae, right? The bacteria which cause pneumonia. It is about the
He,
1928. So he prepared how many strain? He observed
what?
the two strain of bacteria.
Oh,
One was
two, yeah.
smooth colonies and rough colonies. But which one is very harmful?
Rough. No, no, S.
Yes.
Smooth type.
Are you sleeping?
No.
Okay. S type, it is virulent. Virulent means very harmful. Okay. So, firstly, in my injection, this is my injection. Okay. I have taken a, not vaccine, I have taken a virulent. It means it is a S type. Okay. So, if I injected this S type virulent in this mouse, in this rat,
You basically
S
killed
type,
it.
so that caused the pneumonia. And did the
Yeah.
rat will die or remain alive?
Die.
Yes. So now I'm making a reverse mouse. It means die. Okay. So now this rat died. Why? Can you may I know the reason for same?
Pneumonia.
Okay, fine. Now I have introduced R type into the rat. Then what will happen?
It gets jacked.
It was attacked?
It gets no. Okay. Okay. It
Yeah.
gets. It remains
Nothing.
healthy.
Remain alive, right?
Remains
It means
healthy.
nothing will happen. Why nothing will happen?
It's
Why
the
this
rough type,
yes, rough type is virulent or non-virulent?
not virulent.
Yeah. Now, now I I burnt the S type. I mean I have heated the S type. You can say heat killed. So just tell me if you're heating something, so all the bacteria will die. Why we
Not
are
all.
like burning
Hopefully.
the
Hopefully.
yeah, if we are giving a lot of heat like pasteurization, milk, all the bacteria die mostly.
Mm-hmm.
So same thing we are doing. We are like we are doing the heat, like we are giving the heat to the S
It
type. So
purifies the thing.
so yeah, so it means all the bacteria will die. So now I have
Mm-hmm.
given the injection to this third mouse. This is known as the heat killed. Heat killed
Mm.
what? Heat killed S type or R type?
S type.
Because that was already non-virulent R type. So that's why we are giving heat to the S type. Heat killed. So if we are giving the heat killed S type to this mouse, so of course all the bacteria will die. So when we
Mm-hmm.
are giving the injection, so just tell me rat will die or not.
Theoretically, it should not.
So rat will remain alive. Oh my god,
Yeah,
very
alive.
bad rat. Don't say like rat will become very weird. No, no, no. Now rat is not becoming weird. It is remain the same. Okay, just the diagram.
Mm.
Okay, like this. But now, oh my god, this rat is very weird. Okay. Now, so it
What?
means from the heat kill,
It's a fat rat. It's a fat
uh,
rat.
heat killed S type and R type
Mm-hmm.
both are basically making the rat alive.
Yeah.
Now, if you are mixing both of them,
SR type.
if you are mixing both R type and the heat killed S type, so what do you think? Rats should remain alive or die?
Die.
Arre, R is
Alive.
also
Alive.
R strain, mouse remain alive. Heat killed S type, mouse remain alive. But when I injected the dose of both the combination, I don't know why, what happened, but the rat died. Died. I asked the same thing to one student, like, why this rat is going to die? So he said, because minus minus plus. मैंने कहा, don't apply this logic minus minus plus. It is not your maths. Just tell me what is the reason. R was non-virulent. Heat killed S type also become the non-virulent. So how
Well, you don't know
non-virulent?
how they could react if both of them come together.
Oh, so it means something happened.
Yeah.
Something like if your friend is very good, okay with you, and your other friend is also very good, but when both are coming, so you they can't guarantee that both are very good in front of them. Okay, so it means, so we have to find out the reason something happened. Something definitely must be happened. Okay, so now
Yeah.
what I have to say, see, if I have burnt, if I have given the heat to the S type, heat killed S type, so it means it has already died.
Hmm.
Bacteria has already died, so it means definitely it is not because of heat killed S type. It is
Hmm.
not because of heat kill S type, because once, if anyone die, so they can't become alive. So it means this rat died not because of heat killed, but because of the R strain. It means it was
Mm.
believed that R strain has changed its nature. R
Why?
strain has changed because of the presence of S strain.
Yeah,
Just
the
tell me,
heat
just
killer strain.
just tell me, Vishnu, one thing. Suppose if you have a friend, okay, and that friend is very close to you. I have taken this example before also, I know that. If your friend is very close to you, okay, and all things are fine, completely fine, but now you have made a new friend, and now that new friend is claiming you are very much close to her or him. So now that your old friend will behave very weird or not.
Weird.
Yeah, she may feel jealous or he may feel jealous, anything can be. So why? Because now you have new friend. So same thing.
Mm.
Our strain was completely good, non-virulent, but because of the presence of heat killed, that become the virulent now.
Mm.
Okay. So it means finally the rat died. It is because of the R strain material or the heat killed S strain.
The R strain.
So it means, can we say that R strain has somehow has been transformed? R
Yeah,
strain has been transformed something. So this is
but
known.
only in the presence of the heat killed
Yeah, in the
strain.
presence of heat killed only. Yes, yes. Only in the presence of heat killed. It is not alone. Okay. So R strain has changed itself into the virulent or non-virulent.
Virulent.
Yes. So has somehow changed itself. Can we say transform itself?
Yeah. Yeah. Sorry. Yeah.
Okay. So transform itself in the presence of the uh,
Mm-hmm.
heat killed.
Heat killed strain.
Now. We have taken out that strain in our injection. Now, of course, if it is because of this strain, if bacteria is going to die, so it means it has done something with the DNA.
Yeah.
Until or unless I'm not changing your DNA, I'm not doing anything with your DNA, you won't die. Whenever we die, our DNA definitely will die. Okay,
Mm-hmm.
so it means it has changed the DNA of bacteria, this thing.
Mm-hmm.
So it means now we want to find out which is the genetic material.
Mm-hmm.
So now we are going to find out. See, this is our injection thing. This is our transforming principle. Transforming principle means like that liquid, whatever causes the death of the rat. Okay. So now many people used to say that RNA is a genetic material, protein is a genetic material, fat is a genetic material, lipid is a genetic material. Many people used to say that whatever we have, it is a genetic material. Genetic material means which causes the character transmission in the offspring. Okay, so we were actually want to find out. We were very eager. We wanted to find out like which thing is genetic material, like which causes our character to be transferred into the offspring. What is the main thing? Okay, so we
Mm-hmm.
conducted this experiment. Now, just tell me, if this liquid, if this strain was containing the protein, so we took protease enzyme. So protease enzyme break the protein, yes or no?
Mm-hmm.
Nothing happened. So if nothing happened, so it means, just tell me, that liquid, that strain contain the protein or not?
No.
No, because if it contain the protein, so in the presence of protease it will break down, it will structure will rupture,
Yeah.
but nothing happen, it will remain as it is. So because of this, protein can be genetic material. No,
Sure.
no, it doesn't contain the protein na. So if it doesn't contain the protein, so how can you say it
Well,
is genetic
nothing
material?
nothing can be genetic material except chromosome like DNA, right?
This is my injection which contain that thing, both the mix. So whatever we want to find out what it has, whatever it has, it will become the genetic material because it causes the
Oh.
death of the rat. So now we
Mm.
took protease, and protease did nothing. If it contained the protein, so this protein will be ruptured because of the protease, but nothing happened. So does it contain protein?
No,
No. If it doesn't contain protein, so how it can cause the death of the rat?
it doesn't.
So it means protein can be genetic material here? No.
No.
Now we took fat. I mean, we thought like
Lipids.
it contained the fat. So we took the lipase enzyme, but nothing happened. So
Oh, it's a
fat,
carbohydrate
so
thing?
fat can be genetic material.
No.
Okay, fat. So nothing happened. Okay, fine, fine, fine. Now we take like we thought it can be carbohydrate. So we take enzyme like amylase. Amylase break the carbohydrate. Nothing happened. So all the people are saying, "Oh no, this is wrong. This is wrong. They are doing nothing. This theory is bad." Okay, people are saying
Is it
now.
just
Now
DNA?
some people say, "Oh yeah, yeah, yeah. Like it is RNA is genetic material." Many people were crying. RNA is
Mm.
genetic material. Then we took the catalyst RNAs.
Yeah.
RNAs. So when yeah, I'm just leaving it. Don't worry. RNAs.
No, 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 no.
So
That's not
RNA.
why. I
Okay.
yeah. I just
Okay.
remember hearing RNA somewhere.
Okay. So RNAs. So if it contain the RNA, so by taking the RNase enzyme,
It breaks it down,
it
right?
could break that, but
Yeah, it
nothing
breaks it down.
happened.
It's DNA now.
Okay. So now after that we took DNAs. DNAs
DNAs.
enzyme. And when it DNA, so it changes its color.
Oh.
Something happened. It changes its color. It's just
Mm.
broken. Structure will become show some different character. So just tell me that strain was containing DNA or all these?
All of them.
What are you saying?
DNA was the only one that had any sort of reaction. Something happened in DNA.
Yeah, so that strain was
Injection has
containing.
DNA.
Yeah, so DNA is genetic material or the all these?
Everything. No, 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 no. DNA, DNA.
Because DNA causes the death of the
Something to
rice.
happen. Something to
Yes,
happen.
so it was considered DNA is a genetic material. Why? Because that strain doesn't contain protein, doesn't contain lipase, doesn't contain starch, doesn't contain RNA. It
No
contained only
reaction took
DNA.
place.
So it means which causes the death of the rat? DNA.
DNA.
DNA. Okay, so it means the DNA causes the that DNA. It is the DNA which is getting transferred from one generation to another. We can say
Yeah.
that. We can predict that. So it is done
Mm-hmm.
by the Griffith. But many people were saying, no, 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 you are wrong. This theory is wrong. You don't know anything. Why? Because they were saying, I have this skin. This is my protein. This is made up of protein. And my baby also have the protein. So obviously, protein is a genetic material.
No.
My face and my baby face is completely same. So it should be protein. Many people were saying that, but
Well,
many
they're
people were
dumb.
saying no. Whatever you are seeing from outside, it is not from the inside.
Hmm?
Actually, it is DNA. Okay, so in the next class we will discuss about the further topic. Okay, like other scientists came and they also perform many experiments. Okay, so is it clear to you Griffith experiment now?
Yeah.
So Griffith, so who was the first scientist who says that DNA is a genetic material?
Vincent Griffith.
Yes. Okay, so and he worked on which bacteria? They prepared the
The
strain.
s- I can't read it.
Streptococcus pneumoniae, which cause pneumonia.
Strepto
Streptococcus.
oh, strep throat. Strep-
Streptococcus, something like such bacteria which have a chain-like structure.
yeah, the
This one.
beads,
Yeah,
mitochondria.
yeah, Streptococcus, Strepto.
The
Okay,
Hindi thing, whatever you. Yeah,
okay, now got it.
yeah.
So how many strain he prepared in his experiment?
Four.
Four. First simple S, then R, then heat killed S, and then combined.
Yeah,
Okay,
the poison.
so okay, yeah. So and finally he concluded that DNA is genetic. Although people were not satisfied, so they performed many experiment also. Okay, rest of the thing we will discuss in next class and we will follow the next schedule. Okay, I mean new schedule.